Chromosome walking. It is a famous technique used to find and study specific genes on a chromosome. In the first step, the chromosome is partially cleaved using a restriction enzyme. This cleaving produces several adjacent DNA fragments. Together, all these fragments represent the chromosomal DNA. Now these fragments are placed into suitable vectors or plasmids. The plasmids are then transferred to bacterial cells. These cells are allowed to grow and form different colonies. To begin working on the chromosome, we need a starting point. So we select a gene on the chromosome, the sequence of which we already know. Remember, the gene can also act like a probe to find the adjacent gene. For this purpose, we take a tiny part of this gene and label it with P32. This probe hybridizes with the colony that contains the adjacent or overlapping gene. Next, we take out the plasmid from this colony and release the gene from it using a restriction enzyme. Again, we label a small end portion of this gene with P32. Now this probe hybridizes with the colony that homes its neighboring gene. We take out the gene from plasmid, note down its sequence and make its probe. This probe can find out the next gene in the colonies. We repeat this process several times and sequence the genes. This way, we can create a map of the entire chromosome. This map reveals the location of each gene on the chromosome and helps us understand the function of those genes.